Great. So this is Dan Schwartz with MDIC. There's about 23 of us on right now, so I'm just going to do a quick intro here as other folks jump on to the MDICx. I want to thank everybody for joining us for our second MDICx series for virtual patients. Again, these are workshops that feature emerging trends, in this case talking about virtual patient. And with us today, these are going to be outputs specifically on the R-code access and use for the statistical virtual patient model. As for an agenda, Don Bardot will give us an overview of the MDIC virtual patient project, where we've been, and some updates with that. Then we're going to have Sean Belcombe give us an introduction to CRAN and discuss GitHub and actually accessing the code. And then Donald Musgrove from Medtronic is going to go through some case examples where we have applied the virtual patient to various endpoints, outcomes, and trial types. So with that, I will hand it over to Don Bardot of MDIC.
Thanks, Dan. You want to advance to perfect, and you can go ahead and advance to the next slide. So for those of you who aren't familiar with MDIC, although I think most of you are, we're a 501c3 nonprofit. We're a membership-based organization, and our members join us from large and small companies in the medical device industry. We have government members, FDA, NIH, and CMS, as well as a variety of nonprofits that include patients and providers, as well as research organizations. We conduct our work through projects, and we have 6 projects. The output of those projects are put into the public domain through things like this MDICx webinar today. Next slide. Quick snapshot of our current 59 members. Next slide. So I'll give you a little bit of background about our clinical trial science project, a little bit about the method that we'll be implementing through the R code, and all of this has been done through several of our projects at MDIC, but the project that is working on the virtual patient right now is our clinical trial science project, with the goal of advancing regulatory science through innovations in clinical trial efficiency and cost effectiveness. And the virtual patient is a great example of better efficiencies for clinical trials and potentially smaller clinical trials that can all lead to accelerating patient access to new and novel technologies that are safe and effective. Our project is led by board champion Chip Hance, and we have primary contacts out of the FDA for this project, from Owen Ferris and Ken Skodacek out of the clinical trials group within the Office of Device Evaluation. Then additionally, we have project team members for the virtual patient project that span the majority of our membership at MDIC. The timeline for the virtual patient project, we started out in 2014 with this project through the Computer Modeling and Simulation Act project at MDIC. We developed the method, and then we conducted a mock submission. And that mock submission had a sponsor team comprised of a variety of MDIC members and a review team from FDA that were just FDA reviewers. And we went through the Q submission process, the standard process at FDA, but we went through with a mock submission. And we put all of the resultant product on the MDIC website, which you can find at the link on the page. And there you'll see all of our pre-submission materials, the responses that we got back through the review process. In 2016, after we'd realized that we'd, through the mock submission, that we'd arrive at a virtual patient statistical framework that was robust, that we got, that was usable, and that we could start increasing adoption, we started to look at using retrospective analysis to further improve the virtual patient method, increase awareness, and de-risk eventual sponsor prospective use. And that work is also on the MDIC website at the link. And in 2017, right now, we're driving sponsor adoption of the virtual patient method for prospective use in submissions. We have one submission that has an approved statistical analysis plan based upon what sponsors have told us, and we have produced additional content to help support the use of the virtual patient. We have our first publication that was accepted in 2017 and is available at the link. And today you'll hear about the R code that's been created and is downloadable to help folks use the virtual patient method, so you have all of the statistical tools at your fingertips. Next slide. By way of introduction to our motivation to create the virtual patient, we recognized a couple of competing factors that create tension. One is that there is a tremendous and increasing demand for clinical evidence. That's both from the patients, the payers, the physicians, the regulators. Everybody wants more evidence about the performance of medical devices. Even the innovators bringing the products to market want more evidence about how that product is performing. And additionally, we understand that creating that evidence through traditional means, clinical studies, is expensive, and the rate of increase of that cost is unsustainable. In this chart, we're showing 15% per year based upon numbers from a few years ago. MDIC is creating regulatory science to help address both the need to have more evidence more cost effectively. Additionally, if we think about all the evidence that is available, we recognize that our current methods, quite frankly, leave evidence on the table unused. We have increasing availability of evidence through computer models. We have evidence that's available through post-market information, through routine clinical care, and perhaps the lowest hanging fruit, we have clinical information that's available from clinical trials that are done at other parts of the world. Oftentimes, clinical trials are thought of in isolation, but we often have knowledge of how the device will perform based upon first-in-man that may have been done outside of the U.S. and other clinical studies that might have been done in other geographical locations. And so that is an opportunity that we wanted to address, an opportunity to aggregate all of the information that's available when it's exchangeable, when it's of the appropriate quality and fit for purpose. So this prior information is an opportunity that we incorporated into our virtual patient framework. Next slide. The statistical framework that we created is a Bayesian statistical framework, and in 2010, FDA released guidance document identifying the value that Bayesian clinical trials could bring and indicating that informative priors could be used, but at that point in time didn't offer examples of informative priors. And we've addressed that by creating an informative prior methodology where some of the concerns around determining the strength of that prior information could be addressed in a logical fashion. So if we think about the study data that we collect through the clinical trial and all of this prior information, we know that when they agree, we'd like to be able to use all the information, and when they don't agree, we don't want to be using that prior information when it's not exchangeable, when it's not appropriate. So in this way, we recognize that creating a Bayesian statistical framework with what we're calling a virtual patient as a way of thinking about the strength of the clinical trial prior evidence could be a way of creating an informative prior that was very understandable and approachable. So the MDIC working group created that method, and the way that the strength of the prior is controlled is through a discount function. When the prior data and the current data are in agreement, the prior data is allowed into the clinical trial as virtual patients, and when they're discordant, it's not. Next slide. So this is a little more information on the discount function. The discount function is something that's agreed upon in advance, and you look at the performance and the characteristic of that discount function as part of your statistical plan negotiation. If your prior data is exchangeable, mature, and credible, it's reasonable to have a less conservative discount function, where the prior weight, when the agreement is good, is more. More of the data is allowed into the clinical trial design. Next slide. So the discount function is incorporated with a Bayesian power prior to create the virtual patient statistical framework. And this happens in a logical three-step process. So once the trial is underway and either complete or at interim analyses points, you compare the prior information with the current information. That determines the amount of agreement, the concordance. From that, your next step is to compute the strength of the prior information based upon the pre-agreed upon discount function. Then you combine that prior information with that current information to create your total information for the clinical study. And that's graphically depicted on line item 4 in this picture, where the red is your prior information, the blue is your current clinical trial information, and the combined is in the dashed lines. Next slide. So MDIC, after creating this method, working through the mock submission, we engaged sponsors through a working group to conduct retrospective analyses. Those retrospective analyses asked the participants to take a look at existing clinical trials that they had completed to find 2 clinical trials, one clinical trial that would be the prior and one that would be the current, and to ask the question, if you'd used the virtual patient statistical framework, what would it have been beneficial to you, and what would we learn? And the table shows the 4 completed retrospectives that were completed in 2016, and you can see that we were able to include virtual patients in each of these clinical studies, and the total amount of virtual patients accounted for a range 50% at the maximum, and in a 2-arm vascular example where the discount function was the same for test and control, we had a 16% savings, although we recognized that if we picked a different discount function for test from control, which would be a reasonable choice, the savings may have increased. The 2 orthopedic examples listed 36% and 25% for the virtual patients. And these were for retrospectives that hadn't truly been designed with this intent, so this is a good indication that virtual patients works, the savings is real, and it's ready for prime time, ready for prospective use. So next slide. So in 2017, MDIC has conducted a series of presentations to engage the community and raise awareness around the method for virtual patients. We have an upcoming presentation at the Joint Statistical Meeting this summer. And we've hosted now, today is our 2nd online MDICx event this year, talking about the virtual patient. And today we have the exciting opportunity to release the R code that's been built, the R code that can be downloadable and has the statistics necessary embedded in it for the virtual patient framework. We've picked this as being an open source option that will be downloadable by everybody. So we decided that would be a great first step in increasing the adoption and usage of the virtual patient. Next slide.
Thank you, Don. So with that, I will now turn it over to Sean Belcombe and Donnie Musgrove, who will walk through that R code release as well as case examples using it. So Donnie, I'm just going to, you should see I'm passing you presenter rights.
Great.
All right, so as Dawn had mentioned before, our goal was to make this project open source. And so we have a couple of tools that we're using to reach that goal. CRAN, which is a package manager for R. It's basically the de facto package manager. And we're using that for the release versions of this code. And GitHub is where we store our development version. And we've made that open to the public so that people can look at the code. There's transparency to it. They can actually just download the code as it's being developed and use it. And so this really, this helps the project in that there's a lot of good code out there, a lot of good software, but it can die on the vine if it basically stays sort of hidden in maybe some lab group's web page or some professor's web page. So it's a good option to be using these central hubs to distribute and promote your software. So first off, if you've used R, it's very likely that you've made use of CRAN. You may not know it, but whenever you use the install packages command, if you're using anything beyond the base functions that are provided, you're using CRAN. CRAN is a package manager that handles dependencies. In our case, because CRAN holds libraries for R, it handles the dependencies of other libraries that are required by any given library. So it handles the downloading and installation of all the libraries necessary to use the functions in a given library that you want to use. So our package lives at the top there. Every library in CRAN is given its own home page. There are links to the documentation for the library. You can download the tar file of the source code, binaries, et cetera, et cetera. So CRAN is this volunteer network of people who are playing a part in the curation of distributing R code. And so the CRAN webpage, you can go and learn more about CRAN. I think there's about maybe 800 or so libraries at the moment. 10,000, okay, much better. Sorry, I undershot that. So yeah, CRAN is a big deal. And one of the other options that'll kind of lead us into the next step here is that CRAN actually makes the source code for all its libraries available in a read-only format on GitHub. So if you don't want to download tar files, you can take a look at the source code just in your web browser, et cetera. Or you can, if you use Git, you can clone the versions of those packages. So next slide. All right, so our first release was March 22nd, so it hasn't been around for very long. We've done a few updates since then, but the CRAN logs library allows us to take a look at downloads from CRAN, so we can take a look at how popular our package is, et cetera. And we've had a few minor updates, and some major ones. So we can see that there's a spike whenever we do an update. But I've compared the package to other similar Bayesian packages. And I mean, we're not ggplot2, but we get, for a really new package, this is pretty good. So next slide. All right, so like I said before, documentation is linked to on CRAN. So if you need help, you can access manuals directly on CRAN, there's PDFs. You can take a look at the vignettes, et cetera, et cetera. These are all available in the package within R as well, but if you're not haven't installed the given package that you're looking into, it's all available on the website there. And also, we have the option, if you run into a bug or you have questions or need a new feature, we're directing you to our GitHub page to promote those issues. So you can take a look at that. Next slide. All right, so that leads us into GitHub. So you may have used source control software like SVN or Git. Some people are familiar with this. It's just a way of pulling down, hosting and pulling down code that you're developing, and it keeps track of any changes, et cetera. So yeah, you can obtain Git from the website there. And what GitHub provides is a central hub for Git repos. So you can obtain our development version. It's actually attached to my username right now, but that may change. But you can go to the web page there. If you know how to clone something with Git, you can clone the package in the state it's in, or the development version of the package anyway, and obtain the code, and you can build it within R. So the DevTools package actually provides a function that's very akin to install packages, where you can instead install directly from any GitHub hosted library. So with the install GitHub. So that's an option. This is a little extra, but if you're using OSX, I've run into issues. That little piece of code there will clear up at least one issue I've run into as far as installing the development version directly from Git. The other option is that if you just want to use Git manually in the terminal, you can just clone the package and then use R command build to build the package and use it. That's that. So this slide is kind of an addendum. So in the process of developing this for QC purposes, we have used a lot of different services to make sure that the package is in working state. RStudio provides ways to do build testing, build checking. On top of that, CRAN itself, because it's a curated project and they're very concerned about quality control, use an extra service called WinBuilder. We've noticed that we use WinBuilder, and if we don't get any notes, no errors, we get releases through faster. So they really depend on that. Again, DevTools allows us to use a simple function to submit to WinBuilder as well. But in addition, for the GitHub page, for our GitHub repo, we're using Travis CI. Travis basically builds a virtual environment, pulls down any necessary R libraries, et cetera, and attempts to build the package. It's a great way for QC to make sure that at any given state, whenever we push a commit to the Git repo, it tests it and makes sure it's okay. If it doesn't work out, those little bonus shields, they're called, will go red and tell us what's wrong, et cetera, et cetera. So it's a nice way to make sure that everything is in a working state. It also communicates to the users, if they go to the page and they want the development version, what state is it in now? What's the snapshot look now? Do I look like now? Do I want to pull this down at the moment, or do I want to wait for the next commit? But the same testing infrastructure, this is the way I've used it, is using it for just the practical aspects of getting things to compile, make sure the documentation's intact, et cetera, et cetera. But it can be expanded, the same infrastructure can be expanded on for things like validation, et cetera. So I think that leads into the case example, so I'm going to turn it off to Donnie right now.
All right, thanks, Sean. So I'm Donald Musgrove. I'm going to now talk about case examples of our package. So like Sean said, our package is called BaysDP, and we currently have three main functions that we use. They're called BDP binomial. This handles binomial count data. BDP normal. This is normal mean data. So if you're able to summarize your data using mean standard deviations. And BDP survival. So this handles survival outcomes. Now we can also handle all kind of permutations of these analysis types and data sources listed here. So for a single arm, this is the case, say an OPC, where we only have treatment data. And for a two-arm analysis, we have treatment plus a control arm. Now for the data sources, and at least in this package, we're calling it current and historical data. I know Don referred to the historical data as prior data, but we view it similarly in this package. So yes, I'm going to have about 4 case examples I'm going to go through. So I'm going to start off with a single arm trial with a binomial count endpoint. So in this example, we're going to assume we have similar or fairly similar event rates between the current and historical data. So here, we've got our historical data in hand. We've had 28 events. in about 450 patients. And a priori, this is an event rate of 0.06 in our historical data. Now for our new data, or our current data, so far we observed 8 events in 200 patients. So a priori, this is an event rate of 0.04. Now just to kind of illustrate, so you get an idea of what we're doing, if we were just to simply combine this data into one data set, we would have an event rate of 0.06. Okay, but we'll see how our approach handles this. So to model binomial count data, like I said previously, we use the BDP binomial function. Here, the inputs are listed. So the y sub t, so these are the counts for treatment data, current treatment data, so it's 8. And then n sub t is the sample size of the current treatment data. Now the next line, I have a 0 attached to the y. So in our package, we let the 0 denote the prior or historical data. Great, so we run it, and a second later, we have our results, and we can apply a summary method. So each of our functions has the default R summaries, or the default R methods that include summary, print, and plot. And I'll show examples of each of these as we go on. But now, so we have summary of this particular fit, and this is modeled after binom.test in R, if you're familiar, which is a binomial test. So the results are presented, perhaps they may appear out of order, but we really just follow the binom.test function. So first, we see the first 2 lines. This is just the data we've input, so the current data, historical data. Now the third line, we see the stochastic comparison. So this is the first step in this approach, or the first step. And we see this value of 0.85. So this somehow represents the concordance or discordance between the current and historical data. So in this particular case, this suggests that the current and historical data are fairly concordant, and this is reflected on the 4th line where we see the discount function value alpha. That's a value of 0.70. So this discount function alpha represents the weight, or perhaps the amount of the historical data we're willing to use. We also have presented a 95% interval here and what I'm calling the augmented sample estimate. So now the probability of success listed is about 0.055. So this is the current data augmented by the historical data with the given weight, and this is our new rate. So recall before we had a rate of 0.04 for our current data, 0.06 for our historical data. Now this 0.055 is really close to the historical data, and this makes sense because we had a large sample size in the historical data, and we're given a large amount of weight. Okay, and what I just said is reflected in this plot. So like I said, we have plot methods for our functions, and in particular, you can assign a type here. So you can see on the left, we have plot fit, so that's the object, and then the type equals posterior. And this shows you the current data rate, ignoring the historical data rate, and then the augmented data. So the augmented data is the current data augmented by the historical data. And we can see what happens visually. So our current data rate was 0.04, historical data rate was 0.06. We combined the two, and now we've got a value of 0.055, and this density reflects that. Another type we have in our plot method is called discount. And this lets you visualize the discount function that we're using. So in particular, I ran this with the defaults, and the default is a Weibull cumulative distribution function with the shape parameter 3 and a scale of 0.135. So these are just held fixed. And actually, in a few slides, I'm going to have examples of different scales. You can see how this changes. But one thing we can see is this symmetric around 0.5. So values of 0.8561, which we observed, will give the same weight as a value of 0.1439. So you can tell then that 0.5 is 100% weight, and as we get away from 0.5, the weight starts. Okay, so now seeing this example, you may think that that 0.04 and 0.06, these are quite a bit different. Perhaps I don't want to give 70% weight to the historical data. I want to give 10% weight. So we have functionality that allows that. So here we can set a max value, so alpha max equals 0.1, 10%. And we can actually have the analysis fix it at 10%. So we have another input, fix underscore alpha equals true. So this means that no matter what the stochastic comparison, the discount function value alpha will be 0.1. And that's reflected on this slide with the summary. So again, like I said, we see the stochastic comparison is still 0.8561, but we ignore that and we just use the fixed weight of 0.1. And now the 95% interval and the probability of success reflects that. So now the 95% interval is much wider than it was before, and now the probability of success is closer to the current data. And I've got a side-by-side comparison to help you visualize this. So on the left, alpha equals 0.1. We're giving very little weight to the historical data, so the posterior really reflects the current data. On the other hand, with alpha equals 0.7021, on the right, we see that the posterior is somewhat in between the current and historical data. I apologize, I've kind of violated the cardinal rule of having different y-axis limits here. But if you look at the green and blue curves, they are exactly the same. It's the red curve or the posterior curve that is pushing up the y-axis. Okay, so now I'm going to move on to the two-arm example, and I'm going to illustrate this using the normal mean endpoint. So here now we assume that we have historical data on both treatment and control, and we have current data for treatment and control. Now for simplicity, I'm going to assume that we have the same standard deviations and sample sizes for each of the arms. And the only difference really is the control and treatment. So here, the historical treatment has a mean of 50. The current treatment has a mean of 45. So a priori, we know that the current treatment is much smaller than the historical treatment. Now the current and historical controls, though, they're going to have the same distribution. So they're both going to have a mean of 55. Okay, and then to input this into our function, into our package, we use the BDP normal function. And this is probably making sense based on what I described earlier, but the mu, sigma, and n, these are the mean, standard deviation, and sample sizes. The underscore T, that denotes treatment, underscore C denotes the control, and the 0 denotes historical or prior data. Great, so again, I'll run this with all other values, other inputs we have as defaults, and I will run a summary method on this function. Now this function, the summary here is designed to look like a two-sample t-test in R. So again, we followed kind of the layout that the t-test method used, so it may look out of order. Okay, so the first 4 rows, this simply shows the data that we've input into the function. The next 2 are the stochastic comparisons for the treatment and control arms. Now recall the treatment arm current data had a mean of 45, and the historical arm treatment data had a mean of 50. Now based off of our discount function, this gives a weight of 0 on the historical data. So for this example, we essentially ignore the historical data, historical treatment data. Now for the control data though, as we saw, both historical and current and historical had the same mean of 55. So they did a stochastic comparison, the p-hat here, essentially 0.5. We can't really discern the 2. So this results in a weight of 1. And then we have a 95% confidence interval that we're calling, and this is centered around -10, and it's very, very tight around -10. And this makes sense because if we look at the last line, the mean of the treatment group and the mean of the control group, we have 45 versus 65. Okay, so now looking at the method, the plot method for our fits and posteriors type, we now get 2 panels, one for the treatment arm and one for the control arm. So now we can see why the weight did what it did for the treatment group. the current and historical data are almost completely separated, and the posterior falls right on top of the current. And this, obviously, this makes sense because I described that we ignore the historical data. Now for the control arm though, again, this also makes sense. The historical and current data have the exact same name. In fact, they have the exact same distribution. So it's intuitive that the posterior would have the same distribution as well. Okay, and our discount type for the fit here also shows two panels, one for treatment, one for control. So on the top, we can see the zero stochastic comparison resulted in 0 weight. And that's in the bottom left corner of the top panel. Now in the bottom panel with the control arm, we essentially estimated a stochastic comparison of 0.5, and this resulted in an estimate of 1 for our weight of the historical data. Okay, so I'm going to move on to our final two examples, and these are good. Both can be survival endpoints, single arm and 2 arm. This is by far, I think, the most interesting part of our package. So for the single arm example, I actually need to simulate data, so we expect a data frame as input. So here I'm going to assume that our historical treatment has a hazard rate of 1/10, and our current treatment has a hazard rate of 1/15. And just for simplicity here, I'm going to assume there's no censoring, but I do want to point out that this function can handle right-censored data. It's actually what I'd expect. And now for single arm, we are interested in estimating the probability of survival at some time point. Now for this example, I'm going to say we're interested in estimating the probability of survival at 10 years, but this is totally up to the user and what the clinical trial is demanding. Okay, so now to generate data, I'm simply simulating from an exponential distribution with the hazard rates listed, and just kind of the true or 10-year survivals or the theoretical survivals are 0.37 for the historical treatment and 0.51 for the current treatment. And to input all this into our package, we use the BDP survival function, and this input's a little different than the other two functions. This expects a formula, and the formula, if you're not familiar, this requires a survival object. So that is that little serve right there, serve time status. So this comes from the survival package, and we've kind of just emulated what that package does. We also need to input data, so we need to generate a data frame, and then you need to input the survival time of interest. So here I just give an example of what the data frame actually looks like. In our package, we need the column names to match exact. So we need columns with names time, status, treatment, and historical. And the inputs are, I think, fairly self-explanatory. Okay. So we've run the BDP survival 1 arm, and we can apply our summary method. And now this summary is modeled after the serve fit from the survival package, and that's primarily concerned with generating a survival table. And that's exactly what we've done here. So first, I'll talk about the top 2 rows. This shows the stochastic comparison between the current and historical treatment data. And we've got a measure of concordance about 0.10, 0.11, fairly low, but this actually resulted in a weight on the historical data of 0.4. And then as we scroll down, or as we go down, we get the survival table. So this survival table, each row is generated on each unique time point in the current data, current treatment data. So for instance, this very first one at time 0.4322, the number at risk were 50, so I simulated 50 time points. There's a single event. This decreased the survival to about 0.97. We get a standard error estimate, and then lower and upper intervals. And all of this is computed on the augmented data, so the current data augmented by the historical data. Now I introduce a print method. So this is one of our only functions that actually has a print method that appears different from the summary method. But here we get an estimate of what we requested. So we requested a survival time of 10 years, so we request survival probability at 10 years. And in this case, it was 0.441. And this is pretty much in the middle between 0.37 and that we saw for the theoretical distribution and 0.51 for the historical data. And now we see the lower and upper intervals here. Okay, one thing we do here is a little different than our other functions. With survival, we can generate survival plots. And here, this shows somewhat intuitive results. We look at the top red curve, that's the current data. It's well above the bottom green curve, which is the historical data. And now the posterior, or the current data augmented by the historical data, is the blue curve, and it falls in between the red and green curves. Okay, now I just want to take this time to quickly talk about this discount function and what it looks like. So in the middle here, we have the Weibull shape and Weibull scale. So this can be changed by the users. The Weibull shape, the default is 3, and the scale default is 0.135. And that's what I've been using in these examples so far. And actually, we recommend a shape of 3 because it gives this sort of the general curve that you see below. So when the scale is small, about 0.05, the weight shoots up fairly quickly to 1 with respect to the stochastic comparison. So that means for a fairly small stochastic comparison, in this case 0.1, if we were to use the scale of 0.05, we would estimate a weight of 1. Now we may have reason to not want the weight to shoot up so quickly, so that's why we recommend our default of 3 and 0.135 in the middle, and that results in an alpha of 0.4. But you can be even less conservative and use a scale of, say, 0.2, and the steepness of the discount function drops off. So now, in this case, on the plot on the far right, for the p-hat, or the stochastic comparison of 0.11, we would estimate a weight of alpha 0.15. Okay, now move on to the final example here, and this is our 2-arm survival endpoint. Now, this is kind of the point of difference with all the other functions. The 2-arm survival is based off of a hazard ratio between the treatment and control arms. Now, with the 1-arm survival, this was based off the probability of survival at some user endpoint. I just want to make that clear. So in this particular example, I'm going to use the same data as previous. So our historical data had a hazard rate of 1/10, and our current data had a hazard rate of 1/15. Now, we're just going to introduce current control data. So we're not going to have historical control data here. And we're going to assume that that has a hazard rate of 1/12. And again, for simplicity, I'm going to assume no censoring. And now, like I just stated, the interest lies in the hazard ratio comparing the treatment and control arms. The gray box, we see how we would input this, BDP survival function, again, a formula with that survival object, and a new data frame. Okay, so, and just a reminder, right, okay. Okay, so now we've run the function, we can apply our summary method to our fits, and we can see our data input. So the first 2 lines here, this really just shows the current data that's been input. So like I said, I simulated 50 data points, and each one was assumed to have an event that we observed, so no right-censored data. Now the stochastic comparison here, p-hat, is on the treatment arm only. And this makes sense. All we have is a current control. We don't have a historical control, so we can't really do a stochastic comparison on the control arm. Now in this case, the stochastic comparison between the current and historical treatments was 0.04, so very, very small. And this resulted in a weight on the historical data of about 0.03, 0.0299. Okay, so we used that weight value to combine the historical data with our current data, and we estimate a hazard ratio that compares the treatment and the control arm. And that's shown in this table here. So the treatment row, that's really an indicator of treatment versus control. And what's reported is the log hazard rate that compares the treatment and control. So we have a value of about negative 4.692, and then the lower and upper intervals are computed based off of that log hazard rate. Now this might look familiar. This is the same summary method as Cox regression from the survival package in R. If we want to report the hazard ratio, we simply take the exponential of negative 4.692, and we get that in the second column, and that's about 0.0092. So this is very, very small and very, very far from 1, and this makes sense because we know that our current treatment data had a hazard rate of 1/10, while like our current control data had a hazard rate of 1/12. Okay, finally, we can visualize these survival curves, again, using our plot method and a type equals survival. On the top, we can see the survival curves for the treatment group. And what we find now, kind of in contrast to the one arm, is that the current data in the posterior curves are essentially the same. And this makes sense because the historical data did not contribute a lot of weight towards estimation of the posterior. Now for the control arm, may be difficult to see, but there are in fact two curves there, but it's simply the current data and the posterior are the same, so they're plotted on top of each other. Okay, I just want to leave off that the package is under active development, so if you're to visit our GitHub repository now, it's probably different than if you visit it tomorrow, because we're always implementing new things, making changes, trying to improve functionality. Some models that we're going to implement in the future, though, include negative binomial and regression models, so we'll have a regression framework. So far, we've got plans for linear regression, logistic regression, and Cox regression survival. And of course, we're open to further suggestions. So if you saw anything you think could use improvement, or maybe you would like to even contribute, you could contact me. I've got my contact info listed here, my email, donald.r.musgrove@medtronic.com. I can also handle statistical and technical questions. If you have coding questions, or perhaps you have your own R package and you're having trouble getting it submitted, you can even contact Sean S. Baucom at MDIC.org, and he can help you out. Now, I've got listed our CRAN homepage here. Probably easier just to open Google and search for BaysDP, put the top results. Getting help, Sean pointed this out, vignettes, and formal bug reporting. So if you do come across an issue with our package, you can email us, or if you do use GitHub, you can do the formal step, take the formal step of submitting an issue via GitHub. Okay, and that's everything.
Excellent. I want to thank all of our presenters, Don, Sean, and Donnie, for the great presentation today. I'm going to open it up for questions. If you look on your screen, you should have a Q&A box. I believe it's going to be in the lower right-hand corner of your screen. So if you want to put in questions in that spot, I can read those off for the folks, and we can go ahead and get some answers.
Yeah, and thank you very much, Donnie and Sean. That was a really fantastic presentation. You guys have done some really great work. And I'll just remind the audience that our goal here was to help adoption of the virtual patient method, and by providing the statistical packages in R, we're helping lowering the barriers for folks to get in and start using this. So definitely we're targeting our statistical colleagues on this one, but hopefully it makes it very approachable.
Oh, did you want us to answer these questions?
Yeah, can we see the questions on there? I wasn't sure if everybody had the opportunity to see those. So yeah, we can go right through there.
Okay, so I'll just kind of go through this list. So this first one says, what is Balcombe/BayesDP on GitHub?
Yeah, so I'll handle that. So basically, we wanted to have available 2 versions of this package. So any of the release versions get sent to CRAN. But as we're working, as we're developing, we're committing our changes to the Git repo. So basically, GitHub is allowing you to take a look at our progress, what we're working on right now, what the state of things is right at the moment. What we send out to CRAN has some level of stability. What you find on the Git repository is the development version. It may not be in working order, but that's why we have things like Travis CI to give us feedback, whether things are actually compiling, actually able to be used in the state that they're in at any given snapshot. So it's really just giving people the option of either having something that they can trust or having the latest and greatest version of the package.
Yep, and I'll just add to that. Thanks, Sean. I'll just add to that for, I would say, the majority of folks, the only thing that you would ever do is use R and download the R package, the BayesDP R package, and just run from there. But if you want to get into the guts of the package, if you want to take and make it your own and build it into your own code, you can go to GitHub and get all of that information there. So it's just part of the commitment to transparency and openness. It's available for you if you want it, but most folks will probably just use the R interface and download the package from the CRAN. Yep.
Great, and this is Dan. I'll just take one of the questions to make a statement for folks. One of the questions was the availability of slides. So this whole presentation is recorded and it will be made publicly available on the MDIC website. So once we get this recording uploaded, we will make the attendees aware of that. So you'll have the opportunity to view it at any time according to your leisure.
Okay, it looks like we got a question. Why does the loss function drop off at high values near 1? So this is beyond the R package itself. So I see Tarek here if he wants to take the answer.
And I think, yeah, good.
Could you repeat the question? I don't see the question.
All right. Someone asked, why does the loss function drop off at high values near 1? So I believe this means when the stochastic comparison is 1. Right.
Yeah. So it's a symmetric distribution. So the way that the statistical comparison goes is it's a inequality sign. So values that are close to 1 or close to 0 represent statistical difference. So it's kind of like a p-value except like a two-sided. So values 50% is the most the same, like flipping a coin. You don't know which distribution it's from, they're the same. And then if it either goes close to 0 or close to 1, that's essentially the probability that there's a large difference. So that's why it's symmetric, and that's why there's 2 places where you see the differences between the data sets.
Yeah, and I'll encourage all the audience as well. We have both the publication that's available for folks to download. We'll make sure that that's easily linked, associated with the video recording from today. So if you want more information, you can go to the publication. And additionally, last month we had an MDICx that talked about the method from a higher level, from an implementation level. So that would be also a good video to look at for reference, and we'll make sure that those are easily referenced and linked to each other when we post the slides from today.
All right, so I'll read the next question. So someone's asking, in the survival model, can you elaborate more about the model, say treatment plus historical? So the way the model works, it doesn't actually do regression. It just needs to be specified that way so we can essentially know what column contains the treatment indicators and what column contains the historical indicators. So you have 4 columns that you need to input: time, status, treatment, historical. Now, treatment is a binary indicator, so you put 1 where it's a treatment and 0 if it's a control. And historical, you put 1 if it's a historical or 0 if it's current. And that's it. So we don't actually use the data, say, in a regression setting. We just use it to differentiate use it to differentiate the data. Okay. Okay, so I'll read this next one. Not sure what this is asking. So does the discount function mean how much you can save for the clinical trial by incorporating historical data? Right, so the discount function produces a value between 0 and 1. 1 being 100%, so you use 100% of the historical data that you've provided. 0 being that you use 0% of the historical data that you've provided. And you can imagine everything in between, just the percentage of how much of the historical data you're willing to use. Right, the next one, I'll just read this whole thing. It says, it seems the package does not allow to incorporate baseline characteristics of the populations to determine exchangeability. Rather, it only looks at event rates in historical and current. I would say that's correct, yes. Like I said, we do not have this developed for a regression setting yet where we may be able to adjust for baseline characteristics, but that is not something we have done quite yet. Sorry, do you want to add anything to that?
No, simply that is the next step in the package is generalized linear models, which will then allow you to adjust for baseline covariates.
But you could adjust for them in advance and then put them into the package as your data right now.
There's probably some method you could do that would kind of adjust the data prior to input. But I think if that were to be done, I think you would want to just wait for the next section of the package.
Okay, last question. Someone's asking, can you see my questions? And the answer is no.
So this is Dan. I've got another one. I'll just read this one off here. So it says all of those functions, for example, the BDP binomial, are for analysis only, or can they be used for study design perspective to get power, type 1 error, et cetera, et cetera?
I can answer that. Oh, go ahead.
No, so I was just going to sort of reframe that and say, can the tool be utilized to help design a prospective trial is sort of my summary of the question.
So the tool currently just implements the analysis, and if you wanted to get operating characteristics, you'd have to write R code around the packages. And that's kind of a common way of getting operating characteristics, is to do simulations using the functions as the analysis that's going to be happening. But in terms of simulating all of the complexities of the way you want to run your trial, it is not designed for that. We may implement something where it just does a simple clinical trial with no adaptive looks. You know, it simply just says what is the power if you supply this sample size or this sample size. That would be a pretty easy input. But in terms of implementing a whole adaptive trial, that there's too many variables to input, so it wouldn't be probably feasible to combine it with the package.
Great, thanks. And then another question, taking a look at the package does not allow to incorporate baseline characteristics of the populations to determine exchangeability. Rather, it looks only at the event rates in historical and current. Is that a correct statement?
Right, we addressed
got that one, Dan.
Oh, sorry. Next question then is on the parameter for choosing the one-sided or two-sided loss function, or is there a parameter for choosing the one-sided and two-sided loss function? And then part 2 is, as you simulate the survival data using exponential distribution, is there another choice, for example, using the Weibull distribution or piecewise exponential? So that question's in kind of 2 parts.
Right, so the first one, yes, there is a way to choose a one-sided or two-sided loss function. The input is called to_side. You said it's a true or false, it's a logical. You can find details in the documentation if you're really interested. Now for the second question, yeah, I guess this is about my choice of simulating data. Honestly, you can simulate data however you want. I just chose the exponential distribution because it's pretty easy to understand. I can set a rate and I know what that means, and then I can easily back out the survival at a certain time point. And I just sort of did that for simplicity. But yeah, of course, I could use a Weibull distribution. I could use a piecewise exponential distribution for simulation purposes. Now, I do want to clarify, though, the underlying model in BDP survival is a piecewise exponential distribution. Just want to make that clear. Okay, so now question here, where can we access the source R code? There's several ways to do that. One would be to go through our GitHub repository that we've linked, and you can look at our R code there. Second way would just to be look at CRAN's GitHub repository and find our code there.
And a third option would be to go to the CRAN page and download the tar file and then extract it. You can take a look at the source there because the source tar is provided by CRAN.
Right.
Great, so I think that might be all of the questions we have for today. With that, anything from the presenters, Don, Sean, or Donnie, that you'd like to add as we wrap up here?
No, you know, I see a lot of familiar names in the audience, so if anyone wants to email me, you know, or if you want to collaborate or perhaps work on this package with us, we're open to that. You know, in the spirit of open source, we would really appreciate collaboration. So please, yeah, feel free to email. We'd be happy to address any questions or concerns that you may have with the package.
Or if you simply have suggestions for functions that you want implemented.
Right.
We did have one last question. We have a little bit of time, so I think I will throw that out there. So how do we adjust the baseline covariates when you compare the current data with the historical data?
So I've addressed this in a previous question, but currently the package does not adjust baseline covariates. Right now, as the package implements these functions, a user would have to adjust the baseline covariance outside of the package and then put in adjusted values into the function. Now, I've also said that we are going to create regression functions, and then this will be able to handle adjustment of the baseline covariates.
Excellent, thanks so much. So as I mentioned, what we will do is we've been recording this session, both the presentations as well as the Q&A, and so we will get that up and posted on the MDIC webpage, send a link out to all of the attendees and registrants. Along with that, as Don mentioned, we will make available the link to the most recent publication, so if folks want to look into some of the details of mathematics behind it, that will be available as a reference as well. With that, I would like to thank our presenters, Don, Sean, and Donnie, as well as all of our attendees, and especially those of you who asked questions. We're excited to keep driving this methodology forward and look forward to future collaborations.
Thanks, everyone.
Thank you.
Thank you.
Thank you.
Thank you.
Yeah.